All right. Well, it's a real pleasure to introduce Aaron Lund here for a keynote address. If you weren't at the award ceremony, Aaron is a Bioconductor Award winner for this year. He's a scientist at Genentech and was a research associate in John Marioni's group at Cancer Research UK. He completed his PhD with Gordon Smythe at the Walter and Eliza Hall Institute in Australia. And he's a prolific contributor to Bioconductor project currently, especially in the area of single-cell RNA-seq. I've had the pleasure of working with Aaron at some very late-night sessions where we are debugging packages on Windows specifically. And we've done some work in ontology together, and we really need to wrap that up. He has worked put tremendous effort into the Bioconductor Python interface and the basilisk package. It's a very exciting development, and he spearheaded the engagement of the Bioconductor project with the Human Cell Atlas. So I'm looking forward to this talk, and we'll have questions afterwards. It's a 32-minute video that I will now start for you.
Hi everyone, my name's Aaron and it's a pleasure to be here for the Bioconductor 2020 conference. Well, technically I'm not really here and this is pre-recorded. Now I should also mention that I only got the webcam working after I finished recording the talk and I didn't really feel like lip syncing through it again, so instead I just recorded a random hour of my day to keep you company. Anyway, I'll be talking about some of the work I've been doing, or rather some of the adventures I've been having with new pieces of software infrastructure for the Bioconductor project. If you're an R package developer, I think you'll find this talk to be right up your alley, but end users can also enjoy the process of seeing how the sausage gets made, hence the title. Right, let's get started. So, software infrastructure is one of Bioconductor's biggest assets. Here I'm talking about stuff like SummarizedExperiment objects, genomic ranges, experiment hub, iSEE parallel, which deal with the nitty-gritty of data and resource management so that the rest of us don't have to. These infrastructure packages make it a lot easier to develop analysis packages to address interesting scientific questions because we don't have to reinvent the wheel every time. And whenever these packages get performance improvements or bug fixes or more functionality, it benefits all of us, like a rising tide that lifts all boats. We also get this interoperability where packages developed by different people can function together in a single analysis workflow because they share the same concepts and data structures. And of course, this makes life easier for the end users because they don't have to learn a new programming paradigm for each package. In this sense, Bioconductor's software infrastructure is the fertile soil that nourishes the ecosystem of community contributions, which is this plant here, by supporting more domain-specific infrastructure pieces and ultimately the fancy analysis and visualization packages that people get most excited about. So I'll talk about three things today: jailbreaking iSEE for custom visualizations, integrating Python into Bioconductor packages with Basilisk, and some fun and games with the DelayedArray framework for large datasets. Now, getting each of these things to work was like making a sausage. The final product looked pretty nice, but it was blood and guts and hard work all the way through. However, it was also pretty fun, and that's really how coding should be. Okay, so the first topic is going to be about interactive visualization. Some of you may be familiar with the iSEE application for interactive exploration of a summarized experiment object. Well, I mean, it's all in the name, right? Interactive Summarized Experiment Explorer, iSEE. Yeah. Anyway, this is a shiny app that provides a multi-panel layout to visualize multiple aspects of a summarized experiment. For example, in this screenshot here, we have one panel to visualize the reduced dimensions, another panel to look at the row metadata as a table, and yet another panel to look at the expression of a gene. The app also has a system for flexible inter-panel communications where you can, for example, brush on a reduced dimension plot and see the response on multiple other panels based on the samples that were selected. We also have features such as code tracking, reproducible state, and for some reason, voice control. It's a pretty flexible and powerful app that I'm particularly proud of because it's fully community driven. So there's no grants, no paid positions, just sweat and tears from passionate volunteers. Now, iSEE is pretty useful, but there are still some pain points when people try to use it for real. For end users, that is analysts and scientists who just want to look at their data, iSEE seems a bit too general, requiring some work to set it up to get the exact visualization that they want. For developers, iSEE doesn't have quite enough entry points into the interface to perform advanced customization, for example, to make entirely new panel types and mechanics. And also, as a side note for us, it was a real pain to maintain 30 kilolines of shiny code. If you've never had to do that, it's for the best, let me tell you. To demonstrate these difficulties, let's imagine what we would need to do to get iSEE to show a standard volcano plot for visualizing the results of a differential expression analysis. We would have to open up iSEE with a row data plot. That's not too much effort. Then we would have to manually set the x and y axes to the appropriate fields in the row data. Okay, that's a bit annoying to have to do, and it also assumes that I had the foresight to log transform my p-values before starting iSEE. And then finally, we would have to manually count the number of genes that are significantly up or down to show those statistics on the plot, and that's just impractical. So as an analyst, I just want a volcano plot. So there must be a better way. To handle this, our solution was to make it easy to create extensions to iSEE in the form of new panel types. This allows developers to write custom panels to support specific use cases, while iSEE takes care of all the boring stuff related to managing the data and passing information between panels. For end users, the result is that application setup becomes very straightforward, as you can see from this simple one-liner that instantly creates an iSEE instance with a single volcano plot. The overall goal here is to convert iSEE from a single application into an ecosystem of applications built on the same framework. This gives developers the freedom to create their custom panels in separate Bioconductor packages, and then users can then mix and match the panels that they like to set up their perfect iSEE instance. The strategy is to allow custom panel types to fit into the iSEE framework as first-class citizens. That is, a custom panel can transmit information to other panels, it can receive information from other panels, it has full control over its user interface elements and its visualizations. These custom panels will have the same privileges, but also follow the same rules as the standard panels that we provide in the core iSEE package. The only difference is that these custom panels are written by someone else, which really fits in with the theme of community contributions building on top of common infrastructure. Right, okay, so that's a nice dream, but how do we make it happen? With S4 classes, of course. In iSEE version 2, we completely overhauled the internal structure so that each panel type is represented by its own S4 class. This allows users to very easily control the setup of the app by just creating S4 objects corresponding to the panels that they want. For example, here we have set up iSEE with one reduced dimension plot where each point is colored by its assigned cluster, plus a table of column metadata, plus a heat map. The nice thing is that under the hood, iSEE relies on the class identity to determine what it should do when creating the interface. For example, we have an S4 generic function called generateOutput that takes a panel object X as its first argument. Now, when X is a reduced dimension plot, the generateOutput function will produce a ggplot of those reduced dimensions. When X is a column data table, the generateOutput function will produce a dt data table widget. And when X is a complex heat map, the same function will create, a complex heat map. And so on. So in this manner, the same line of code in the core iSEE application can support a variety of different behaviors. And this makes it very easy to extend. All we have to do is to write a class for a new panel type, specify how that class should behave when generateOutput and related functions are called, and then boom, it'll work inside iSEE. Another nice thing about using classes is that we can use inheritance to organize our panels. Everyone starts from the base panel class, which is about as general as it gets. Then we specialize into subclasses that do more specific things. For example, a column dot plot that shows each column of a summarized experiment as a dot or as a point in a plot. This keeps on going until we get to the leaves of this tree, which are occupied by the classes that end users will actually end up using. For example, the reduced dimension plot or the row data table. We use inheritance to reuse code that is common to multiple classes. For example, the reduced dimension plot, the feature assay plot, and the column data plot all visualize columns of the summarized experiment as points in the plot. This means that the column dot plot parent class can implement a lot of the common methods across these plots. For example, to create user interface elements to color points by a field of the column metadata. Similarly, the table parent class implements a lot of common methods for all table-related panels in this tree, which avoids the need to implement them separately in each of the subclasses at the leaves of this tree. This strategy makes it really easy to write new panels because we can just reuse a lot of the existing code in the parent classes while customizing some of the visualizations or interface elements to get the desired effect. To illustrate, I'll walk through some practical examples. We'll start with a reduced dimension hex plot, which replaces the conventional scatter plot with these hex bins that are colored by the density of points. The idea is to improve plotting speed for large data sets because we don't have to show all of the individual points. To create this plot, we simply made a new class that inherits from the standard reduced dimension panel, overrode the plotting function so that it would use the geom hex function for visualization instead of geom point, and then we added an option to the interface to control the bin size. And that's it. And this gives us a new panel that we can use in iSEE just like we would do with the usual reduced dimension panel. Now, onto our old friend, the volcano plot. We start with the general row data plot, but we add constraints to ensure that we can only specify row data columns containing p-values for the y-axis and log4 changes for the x-axis. We modify the plotting function so that we can get some nice colors on these DE genes in each direction, and we get automatic log transformation of the p-values on the y-axis. We also report the number of differentially expressed genes in each direction in the legend. And finally, we add some interface elements to allow users to change the log4 change and FDR thresholds used to define DE genes. This gives us a nice looking volcano plot that people can use straight away without any setup. A slightly more exotic panel is used for dynamic marker gene detection. This is based on the row table virtual class, which provides utilities for managing a table where each row corresponds to a row of our summarized experiment object. Here, the content of the table is dynamically generated by performing an on-the-fly differential expression analysis between groups of samples selected in other panels. In this particular example, we perform a differential expression analysis between two selections in a reduced dimension plot to identify marker genes in one of the selections compared to the other. This is all achieved by simply overriding the function that creates the data frame to be visualized in the data table widget. We don't have to write any of the actual rendering code ourselves, and we certainly don't have to write the code to transmit information between panels because iSEE gives us that for free. So I hope you can see how iSEE version 2 really enables deep and diverse customization via its S4 framework. So if you're interested in interactive visualization, this is a great time for contributions, either in the form of ideas for new panels, or even better, pull requests, or even better than that, new packages that implement their own panel classes. Some examples are given in the links below, and all of us on the iSEE development team will be happy to help you out if you're interested in giving it a go. Right, on to the next topic. Here I'll talk about some of our efforts to improve Python integration in Bioconductor packages. So why do we want to do this? Well, the Python package ecosystem contains a lot of functionality that complements what we have in R and Bioconductor. And here I've listed a few examples that personally interest me, like image analysis. Python also has more support for non-scientific programming utilities, as companies working on new software technologies are more inclined to write bindings for Python compared to R. And the general philosophy is let's not reinvent the wheel here, or the wheels, little joke there. To this end, RStudio's reticulate package has been pretty great, allowing people to use Python functions as if they were R packages. For those of you who aren't familiar with reticulate, I've included a little chunk of code here to show exactly how easy it is to use reticulate in an R session. In this case, to perform a PCA via Python scikit-learn package. I just load the Python module into the R session, call the function I want, and that's it. Easy. Now, R already has multi-language support for its packages, so I regularly include C++ code in my packages, and I've occasionally fiddled with C and Fortran code as well. I've also heard myths of people incorporating Java code into their packages. So given that we already have access to reticulate, why can't we do the same with Python? Well, it turns out there are a bunch of reasons. The most pressing one is that we need to assume that the end user has an appropriate version of Python and all of the Python packages installed. This means that if our package depends on a particular Python package, we need to include the dreaded system requirements in our package description. And whenever I see that, I think, oh no, because it means that the user needs to do extra work in order to install the package. And I don't know about everyone else, but I found this particularly troublesome for Python, what with all the different installation options and configurations. So easy install versus pip install, virtual environments, conda environments, where does my Python path point to, and so on. So relying on the end user to manually do the installation means that there's all sorts of things that can go wrong, particularly with respect to installing the wrong versions of Python packages, even after you explicitly tell them what versions you need. Another problem is that different Python packages can have incompatible dependency trees. Fundamentally, this is caused by the fact that a package can specify version numbers other than greater than or equal to, which is what we do in R. So if two packages require mutually exclusive versions of the same dependency, then, well, you won't be able to use them in the same environment. And this is particularly problematic for reticulate, which can only support one Python environment per R session. And by this, I don't mean like one environment at a time. I mean one environment for the entirety of the R session. So once you load in one environment, you're stuck with it until you restart R. This is basically a giant weak point for deploying Python code inside R packages. If I have two R packages that rely on different Python environments, I can't use both of them in the same R session. So to get around these problems, we created the basilisk package. So this is named after the snake in Harry Potter, with the idea being that we'd be able to freeze Python dependencies for use inside Bioconductor packages. Most of the heavy lifting here is done by Conda, or specifically miniconda, which we pull down and install in a local cache to ensure that we have a consistent Python installation in the user machines. Each Bioconductor package then creates one or more Conda environments containing the Python packages that they need. All of this is done automatically, either upon R package installation or by using a lazy mechanism where Conda and its environments are created as needed. The resulting Conda environments are fully compatible with reticulate code, so if you know how to use reticulate, there's not much else you need to learn. So here's an example involving the iSEE sklearn package, where the developer just has to write a few lines of R code to instruct Basilisk to set up a Conda environment with the necessary dependencies. Once this is provided, Basilisk handles all of the Conda stuff so that the end user doesn't even have to worry about it. Conversely, the new Conda environments are all Bioconductor specific, and so they won't interfere with the end user's Python activities elsewhere. Now, you might say, oh, Aaron, you're just writing a wrapper around Conda, and you wouldn't be too wrong here, but geez, this was a lot of effort. So even with Conda doing most of the work, there's so many things that went wrong, especially with these alternative operating systems. So like, Mojave, Windows. Anyway, that's all done, so end users don't have to deal with these problems anymore. As for the other problem, Basilisk allows us to use multiple Python environments in the same R session. Okay, that's not technically true, but the outcome is the same, as it allows each Bioconductor package to define and use its own Python environment without stepping on the toes of other Bioconductor packages trying to do the same thing. So if we look on the left here, we can see the conventional situation with reticulate only. So package A loads in its Python environment into the R session, and that blocks package B from being able to use a different environment. In contrast, we can have a look at the situation on the right, where both packages A and B are using Basilisk. When package B tries to load in its environment, Basilisk will detect that the R session's Python slot, so to speak, has already been filled by A's environment. Basilisk will then spin up a new R process for package B to do its stuff with its environment, and once it's finished, transfer the results back to the parent session. This allows us to use both packages A and B within the same R session. By default, Basilisk is greedy in the sense that it will try to fill the global Python slot if it's available, as this is more efficient than starting a new process. But if the slot isn't available, then Basilisk is smart enough to start a new process and avoid the error that would otherwise occur. This capability ensures that different Bioconductor packages that use Python can work together without burdening the end user with the details of the R-Python interaction. I'll now walk through a handful of examples of Basilisk in action. The first is the previously mentioned iSEE sklearn package from Vince Kerry, which wraps the scikit-learn functionality into R functions. Switching to Basilisk here was an easy two-step process. We provisioned the Conda environment with the Basilisk environment class, and then we wrapped the existing reticular code in the Basilisk run function. Then the end user can just call the R functions like they would do for any other R package without requiring the management of any Python dependencies. Even better, other R packages can call iSEE sklearn to access scikit-learn functions in a reliable and portable manner. There's no need to re-implement their algorithms and reinvent that particular wheel. The next example concerns RNA velocity calculations for single-cell data analyses that are mostly written in Python. So here we've created a Velociraptor package that uses Basilisk to pull down and use the scvelo Python package. Then end users just have to pass a single-cell experiment object to the wrapper function, and they get all the velocity calculations back, nice and easy. So again, there's no need to re-implement anything, which gives me more time to do other things like watching YouTube. Finally, I should point out that we aren't limited to using Basilisk inside R packages. So we can use Basilisk inside an analysis script that uses Python for some steps. In this mock example, Basilisk will set up a Conda environment with the necessary dependencies so that later Python steps will run properly. The cool thing is that this script will automatically detect whether the environment exists, and if it does, it will use it, and if it doesn't, then it will create it. This means that even your analysis scripts can be reproducibly run on another user's machine without requiring them to manually set up the same Python environment. I've even used Basilisk to set up Conda environments for use outside of R because I always forget how to do it with Conda on the command line. To sum up, the idea behind Basilisk is to improve the reliability of Python integration inside Bioconductor packages. This unlocks the Python package ecosystem for Bioconductor applications while taking advantage of Reticulate's capabilities. I'm hoping that Python integration will reach the same level of reliability enjoyed by C and C++ and Fortran code inside R packages. Though of course we don't even have to stop there because Conda is more general than Python. For example, we could use Conda to install other interpreters for languages like Julia and get some integration going there. Packages relying on difficult system dependencies could also use Conda via Basilisk to simplify their installation. So there's quite a few interesting opportunities for further synergies across the Bioconductor project. Okay, on to the final stretch. So for the last part of this presentation, I'll talk about some of the fun I've had with DelayedArray to represent large datasets. The DelayedArray framework was developed by Hervé from the Bioconductor core team, and it's pretty cool. So the idea here is to provide an array interface to large datasets that are too large to store in memory. The most obvious application is for file-backed datasets, where most of the data is stored on the hard drive and pulled into the R session on an as-needed basis. This allows us to perform analysis on very large datasets, even on machines without a lot of memory. Now, it's called DelayedArray because the operations applied to these large datasets are not immediately executed. In the example below, let's say we have a backend that holds our count matrix, for example, in a HDF5 file, and we want to compute log-transformed normalized expression values. We start by creating a delayed array to wrap our backend, And our first task is then to divide the counts by the size factors to account for differences in sequencing coverage across our samples. However, when we perform the division on the delayed array, actually, it will not do the division right away. So think about it. If we did the division right away, we'd need a place to put the normalized values. Obviously, we can't store it in RAM because the count matrix was too large to put into memory in the first place. So if we wanted to do the division right away, we'd have to write the output back to another file, which would be inefficient and use more disk space. So instead, our delayed array will just remember that we requested a division and then move on. This approach is applied to the next steps, such as addition of the pseudo count and log transformation, and all of these operations are delayed rather than being executed straight away. Now, let's say we have a function where we need the actual values of our log normalized matrix. In this example here, I'm using the row means function. So when delayed array encounters that, it will extract a block of the data into memory and apply all of the operations that have previously been delayed. This block is then used by the row means function to, well, compute the row means. In this manner, we avoid loading the entire matrix at once, which allows us to process very large data sets in limited memory. So hopefully it's pretty clear why this might be useful. For example, I can load the 10x Genomics 1.3 million brain cell dataset as a HDF5 backed delayed matrix on my laptop, even though it would have taken 146 gigabytes of RAM to store it as an ordinary matrix. From a programmer perspective, delayed matrices support standard matrix operations like subsetting, multiplication, calculation of various statistics like the row means, and so on. And this means that a delayed matrix can mimic an ordinary matrix to the point that we can use it interchangeably in our analysis pipelines without much modification. Now, how far can we push this? So I've already shown you an example of a HDF5 backed delayed matrix, but what other zany things can we do? Well, let's have a look. In Exhibit A, we have the deferred matrix, which admittedly is not a very good name, but whatever. So this particular matrix uses the delayed array machinery to delay the centering and scaling that is commonly performed on a matrix prior to PCA. The rationale is that if we have a sparse matrix and we center it, it's not going to be sparse anymore. We might not even have enough RAM to hold this centered matrix in our R session. For example, in this example here, if we centered the sparse matrix in the usual manner, this matrix would require 12 gigabytes of RAM to represent. But because we're delaying the centering, our deferred matrix is very lightweight and memory efficient. Another bonus of delaying the centering is that we can take advantage of the highly efficient algorithms that are available for sparse matrix multiplication. Specifically, we can rearrange the multiplications so that we can directly operate on the sparse matrix prior to centering, which is highlighted in this expression by this YA product. This is important for approximate PCA algorithms like randomized PCA and IRLBA, which do a lot of matrix multiplication. Here, randomized SVD on our deferred matrix takes just 4 seconds, which is pretty nice. Our next example is pretty similar. If we want to regress out some unwanted variation, for example, batch effects or other things, we can delay the calculation of the residuals with this residual matrix. Again, this uses the delayed array machinery to avoid actually creating the matrix of residuals. In this example here, the matrix of residuals would fill 12 gigabytes of RAM if we were to compute residuals like a chump, but instead we get this lightweight residual matrix object that requires very little memory to store. Even better, we can extend this efficiency to matrix multiplication, where we can rearrange the sequence of matrix products to get access to efficient sparse matrix multiplication algorithms. In this expression here, I've just reordered the application of the hat matrix so that we can directly work on the efficient YA product instead of having to use dense multiplication algorithms. This gives us a pretty fast IRLBA run on the residual matrix, which is effectively a free speed boost if we want to do PCA on the residuals after some kind of regression. A slightly different use case is the low rank matrix. So say we do perform our PCA and we want to make a low rank reconstruction of our input matrix. This is easy enough to do, in theory, by taking the product of the first L principal component and the corresponding columns of the rotation matrix. However, the reconstruction is dense and might not fit into memory. So instead, we use the delayed array framework to delay the matrix multiplication itself. So the low-rank product is only computed for particular rows or columns upon request, allowing us to mimic a 12 gigabyte matrix with just 6 megabytes of memory. In fact, this is the data structure that I use to report the batch-corrected expression values in the output of the fast MNN method for single-cell batch correction. So that's pretty cool. The final example I'll talk about here is some work in progress with TileDB. So in its simplest form, this is a file-backed data store, much like HDF5. However, one of TileDB's selling points is that it can store sparse matrices natively. This has the potential to be much better than our HDF5 approach, where we have to expand the sparse matrix into a dense matrix because HDF5 doesn't have proper support for sparse matrices. For TileDB, it doesn't have to read and write all the zeros, so that saves us a lot of time when transferring data to or from disk. To test it out, we put together a delayed array wrapper around TileDB that we called TileDB Array, obviously, and some of the early tests are promising. For example, the reads and the writes are several times faster with TileDB arrays compared to HDF5 arrays, culminating in a PCA step that's almost 3 times faster. This is very nice for large datasets where the PCA alone can take almost an hour to run. So I think we're just waiting for the base TileDB package to get onto CRAN, and then we'll have a look at the Bioconductor submission for this bad boy. The really nice thing about the delayed array framework is how flexible it is for everyone. If you're a developer of analysis software, all you have to do is make sure that your software works with the delayed array machinery, and you get to use all of these cool toys that I've just described without any extra effort. On the other side of the fence, if you want to write a new delayed array class with special behaviors, then you can be pretty confident that it'll work with any Bioconductor packages that are compatible with the delayed array framework. So this is an easy way to start to make your packages scalable. For example, my single cell software stack lives and breathes delayed arrays, so it's pretty easy to churn through analyses of large datasets without requiring much RAM. I give a few examples here, and you might be wondering why I analyzed fewer cells on a machine with more RAM, and that's because I need some memory left over to watch YouTube at the same time. Anyway, there's a lot of avenues for future works and more delayed array backends, more file formats, etc. And one particularly cool thing would be to see if we can use the delayed array framework to push more computation to the cloud, which would allow most of the heavy lifting to be done on a server that's close to the data. Right, so that's the end of the tour. So hopefully some of this infrastructure will help you improve your existing Bioconductor packages or give you some inspiration to write new ones. I've provided some links here if you want some worked examples, and really the ultimate goal of all of this is to promote synergies between packages that really make Bioconductor greater than the sum of its parts. Of course, all of this work involved efforts from many people, so fellow members of the iSEE development team, a lot of volunteers for testing Basilisk to get it to work on different systems, and a lot of development effort in the delayed array space. Also my colleagues at Genentech. And to finish, I'll just say, now that we have all this infrastructure, it's a great time to start writing your own Bioconductor packages to do some interesting science. So what are you waiting for? Get out there and start writing some software.
All right. Well, thank you very much, Aaron. We can watch that again, or we can I know there's interest in many of the things that went on in there. Let's take a look at some questions. I am able to see the poll. Does creation of new R sessions for multiple Python environments adversely affect multi-session parallel programming?
It depends on it. So in some contexts, it may well do so. So in some contexts where you have a particularly aggressive out-of-memory killer, that can be a problem. But that would be a problem anyway if you spun up too many sessions and they consume too much memory. I haven't personally encountered these problems yet, because most of the time I'm running these Basilisk functions within not in a parallel segment of my code, and I'm relying on whatever parallelization is implemented in the Python package itself to do that. But I don't know. That's something we'll find out.
Yeah, I would agree that it may not be unique to Basilisk, but yes, we'll find out. Next question. Can Basilisk environments share Python package installations if the version requirements are the same?
Uh, what was the first part? Like, can Basilisk environments share Python environment? Can you say that
Yeah, I
again?
think the idea is that if you have two different packages that have the same installation requirements for their Python infrastructure, do they are they able to share those, or do they need to install them separately?
Right, right. Yeah. I think that's possible. I haven't sort of formally provided methods that allow people to do that, but that's definitely possible because you just sort of point to the different packet, point to the different environment that you want to use in the basilisk run command, and it'll use it. Obviously, though, if you're depending on someone else's Python environment, then that R package that implements it also has to be one of your dependencies. But yeah, that's quite possible. That's something I've thought about, and yeah.
Great. Does this mean that Basilisk totally removes dependency of reticulate, even if it is mentioned in the vignette as a package dependency for some BioC package?
So I don't think so. I think Basilisk's role is really as tooling on top of reticulate. So if you write a Python package, if you write a Bioconductor package that uses Python, you'd still be using reticulate, right? You'd still be using like the import, the various, Py to R or R to Py utilities if you need to do that. It's just that Basilisk as a layer around that to make it easier for you to deploy that package, to deploy your Bioconductor package and to use it in complex environments.
Yep. Here's a good question. How is a deferred matrix different from a delayed array?
Yes. So that's so a deferred matrix is a delayed matrix basically. So a delayed matrix is a delayed array that's two-dimensional. So a deferred matrix is a delayed matrix. It was not a good choice of naming, I've got to say. Looking back on it, I should have called it like a centered matrix or something. Anyway, the deferred matrix is basically a delayed matrix where there's only two delayed operations, which is the centering and the scaling, right? That's basically it. The reason why it's a separate class is because I need to add I need to overwrite the matrix multiplication operator so that it can intelligently operate on the fact that the centering is delayed, right? Because normally, if you do matrix multiplication with an arbitrary delayed matrix, the process that is used is block processing, where each block is realized into an ordinary matrix, and then that is multiplied with whatever you wanted to multiply with. So with the deferred matrix, because we know that the operation is centering, so we can sort of rearrange the math a bit. That allows us to do more efficient things with respect to sort of preserving the sort of efficiency properties of the underlying data, so sparsity, for example.
Yeah, it's very interesting. Can Basilisk use packages installed manually? For example, when you are working on less common architectures or OSs?
That's an interesting question. I've seen various people that I know of have done that definitely for their local installations. I don't know how easy it would be to make that into a deployable Bioconductor package. So if you wanted to deploy it, obviously you got to pull the code down from somewhere. And right now we pull the code down from either Conda or pip or PyPy basically. I trust those two repositories, right? But if you wanted to deploy a Bioconductor package and you had just some random Python code, right, that you wanted a random Python package that you wanted to include there, you can definitely do that locally. Don't know how you would do it in a sort of Bioconductor context.
Yeah. Now, here's a question from me. I guess it's kind of buyer beware, right? I mean, you don't do anything to see whether your Python dependency is one that has been deprecated or, needed an update or whatever. Any thoughts about how one could have some sensitivity to whether they're using the state of the practice Python infrastructure?
At some point in time, early on, yeah, I tried to do something like that. I definitely tried to make sure that the all the versions of the Python dependencies agreed with each other, for example, and were up to date with uh, 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 the latest distribution from Anaconda. So at the start, I wasn't using Miniconda, I was using Anaconda. So I just wanted to make sure that every package that you requested was sort of consistent with the Anaconda requirements. But at some point, I stopped fighting the system, and I was like, okay, I'll just let Conda handle the dependency help. So it was just easier to let Conda handle it, and Conda does a pretty good job. But as for the other part of the question, which is the sort of keeping things up to date, that's not a decision that Basilisk can easily make for you. I think that's more of a decision that you make, that you say, okay, I'm going to take my Bioconductor packages that use a particular version of a Python packages. I'm going to change the version of the Python packages, and then you are responsible for also changing the version, the interface code or the changes in output that come with that. So that's more of a I don't have any clear idea on whether I could suggest updates for you, because a lot of times, for example, even Conda's like a bit behind, like PyPy, for example. I don't know. That might be something we could look into, but as of now, it's really just a tool. Like that part of Basilisk is really just to allow you to sort of pin these versions down to make sure you're getting consistent behavior across different platforms.
Yeah, I wouldn't make more work for you there, but maybe something like a Basilisk valid could be something that somebody could contribute, potentially as another package or as a pull request to Basilisk. Yeah. Okay, let's see. So will all these updates be pushed into the Oscar book?
Um, so Oscar uses quite a few, like Oscar doesn't directly use some of these, right? So you'll never see me really talk about it in the Oscar book. But under the hood, they're actually being used under the hood. So for example, as I said, the fast MNN function uses the low rank matrix. By like it just that's what it returns. So you don't notice it, but it's also the reason why your computer doesn't crash, because of you're trying to eat a 20 gigabyte matrix. Some of the various batch correction functions use the residual matrix, right? So again, that's something that they just do it under the hood. That you don't even see it happen. I've in the trajectory chapter that has eventually gotten some work done on it. We're putting in the velocity, this sort of RNA velocity stuff that uses SE Velo under the hood using the Velociraptor package. That name I'm quite proud of. That's a pretty cool name. But even then, right, you don't even notice that you're using, like, the idea is that you won't even notice that you're using Python. Like, just like you don't even notice that, you're using C++ when you run other R packages. It just sort of, you just get the R interface to it, and it just sort of works under the hood. So yeah, they'll make their way to the Oscar book, but you won't see them sort of up in lights, right? Because it's sort of like the infrastructure that makes the magic happen.
Yep. Okay, we'll have two more questions, I think. The first is, where is the best resource, simple for beginners, to use delayed array?
To use delayed array. Okay, so to use delayed array as an end user, you actually don't have to do too much. Okay, all right, let me answer the actual question. So the simplest resource is, I would have thought it would be the delayed array vignette. I'm not actually sure. I haven't read that in a long time.
How about, did you watch Pete's workshop?
No, I was asleep.
Uh-huh.
Go back to that. Because it was 5 a.m. in the morning, Vince.
Oh, come on. So I'll answer that question then. Go to Pete's workshop and listen to the music that he plays, and try some of the exercises. Because I think both as an end user and as a person who's thinking about development, you can get information from that. But as you say, you rack your brains to say, where's the document? Maybe the vignette is good enough. Maybe you need something
Oh,
else.
yeah. Probably the best documentation is in Hervé's head, right?
Yeah.
So,
Those of us who have been there and have implemented delayed array.
yeah, that's right. At some point, I was thinking that, someone, maybe me, right? I don't know, but someone, we should write a book about how, the sort of what we felt, what we encountered when we wrote these backends. Because once you write it once, it's actually pretty. It's like wow, that was easy, right? So yeah, that's something on the
All right.
horizon.
Now, this is probably the final question because it's sort of going at the high level. Thinking of 10 simple rules, can you contribute a couple of rules for new developers?
New developers, wow. Um, haven't been that for a long time. Hold on, let me think. New developers. 10 rules, huh? I guess I can just give a couple. Um, well, one of them is to not reinvent the wheel, right? That's like, there's a crazy amount of wheel reinvention that goes on. And I get it, right? I get it because, you're young and you want to, you think S4 and S3 and, all this object-oriented programming is like the bee's knees and you just want to write, your own stuff to do everything. And that's actually fine from a sort of learning perspective, right? It's fine to do that. But, when you actually want to get some work done, whatever you've done, there's probably someone who's done it much better with more eyes and that's been more tested. So spend some time looking for those. Like for example, when back in my day, right? Back in my day, when I was young, I thought I'd write a version of the SummarizedExperiment, right? For handling iSEE data. And it took me 2 years of picking it apart to eventually replace it with the proper version, right? So I mean, it's like these decisions that you think are, with your sort of custom classes and custom methods that you think, are the best thing ever when you write them. In hindsight, they usually turn out to be, there's usually someone else who something else that turns out to be written better and is faster and more efficient. Usually all of the above, actually. So yeah, just spend some time, just like not coding, right, and thinking about, what you should be doing, and looking for like solutions that are already there, especially with like infrastructure pieces like data structures and so on. And then, I had another piece of advice, but I've forgotten it now. Yep, no, it's forgotten.
Well, I'll throw one in. I'll throw two in. One is don't go it alone. Talk to somebody. I don't know how often you do that.
I'm always on Slack. So
Yeah.
Slack's my Twitter, right? Because if I had Twitter, I'd probably be fired. So I post stuff on Slack instead. So definitely talk to people. Yes, the mailing list can be a bit intimidating. Like the BIC Devel mailing list can get a bit intimidating because you have to like mail people. But Slack's like, you can put emojis there and no one cares, right? So, Slack's pretty nice for like informal chats. And, if you want sort of immediate feedback on some crazy idea that you've got, it's like, you can just throw it out there and see who bites, right? Yeah.
Yeah.
Um,
I say don't go it alone would be one of my rules. And then the other one would be to read a book called The Mythical Man Month. You familiar with that?
I've heard about it. I've never read it myself. I've never read it myself.
So it's basically going way back to software architecture for the VM370, and the idea of your ability to predict just how much work it's going to take to do something is probably not where you think it is. It's going to take a lot longer.
Yeah. Yep. That's definitely true. I mean, it's good that I have so much free time on my hands because it doesn't really matter to me. It's I should spend more time on it. But yeah, that's right, because sometimes I think, you know what, I can knock this feature off in this evening, and I don't. So that's things take longer.
You need the long view, and you've taken it, and you've got some definite, some key contributions, but it certainly is a lot of work.
Yeah.
We thank you for that, and the video should be watched again by everybody, um, just to get it all under your belt. I don't see any more questions in the poll. Maybe you have some questions?
There was one question, Vince, but it was more of a comment than a question in the chat.
Oh, in the chat. Okay.
Yeah, it was from Lambda Moses,
Oh, yeah.
Moses,
Okay.
9 minutes ago.
I think making it hard to use random Python package not on Conda or pip in Bioconductor packages makes sense. Just like Bioconductor packages can't have R dependencies that are not on CRAN or Bioconductor.
Yep. I also agree. But I mean, some like, you know, sometimes, like, definitely if you're just using it locally, you know, basically the user is responsible for what's going on there. So, you know, it's all fair game. But at least in a package deployment context, yeah, that's yeah, random stuff on GitHub is like, yeah, there's no way I could let that into production, right? It's you know, who knows if it'll be there tomorrow, right? It's like,
All right, folks. Well, I think we are at the end of this session. We need to get ready for the next one. Thank you all for participating, and thank you very much for Aaron, everything you've done.